Greetings, everybody. Good afternoon. How you doing? Welcome along to this Let's Talk About COVID Q&A. I'm your friendly host, Gareth. I'm going to be with you for the next 45 minutes or so. I'm one of the science presenters at BBC Radio. You can hear the radio voice there. And I'm also one of the lecturers in science communication at Imperial College. And seeing as this is an Imperial event, seems entirely appropriate. So we're going to get straight in and talk about vaccines, almost straight in, but it's better just give you a little bit of an update on how it's going to go today, what the format is. You are watching on YouTube. Welcome along, by the way. We've been looking at the chat leading up to the event. It all looks very friendly out there at the moment. So one thing I want to say is let's keep it that way. So no swearing, no being horrible, no trolling. Otherwise, we'll ask you to leave. That's on the downside. But on the upside, we really want you to leave any questions, especially questions. I was going to say comments, but most of all, we want questions because we have two top experts who are involved in the Imperial COVID-19 trial who are here to answer your questions this lunchtime. So post them up in the comments. Get your questions in nice and early because we do have a team of moderators, so give them a bit of time to sift through the best questions, and they're going to end up those questions on the panel in front of me, and I'll put them to the experts. So that's how it's going to go. So let's jump straight in and meet our experts. First of all, Anna Blakeney, who's a Mary Curie Fellow working on the RNA vaccine for COVID-19 at Imperial College. So the floor is yours. The screen is yours, Anna. Just tell us a bit about yourself.
Yeah, hi everyone. I'm a research fellow at Imperial College in the Shattock lab. My background is in bioengineering, but I work generally on the formulation of RNA vaccines. And I'm really excited to be here to talk to you all.
Great. So we're going to hear more from you as we go along, Anna, but let's also say hello to Paul Mackay, Senior Research Fellow working on new vaccine candidates and prototypes, very involved in this trial as well. So tell us a bit about yourself then, Paul.
Hi, my name is Paul Mackay, and I have been researching the human immune system for my whole scientific career. And currently my main focus is how to make these vaccines better and safer. And we've been working on the coronavirus vaccine since January, and we hope to be able to share some of our insights into that vaccine.
Yeah, as we'll be hearing. So before we jump into the Q&A, we're going to be asking you a question, dear audience, dear viewer. So we have a poll, and we want you to respond to that poll. So just go to menti.com. There you are. You can see it on the screen. Go to menti.com and use that code 360331. 360331. You can see it on the screen down there. And basically, we're asking you about how much you used to think about vaccines. We talk about them all the time, don't we? Everyone's just thinking about vaccines all the time. But how was it before all this kicked off? So it'd be really interesting. That'll give us a sense of the before and after, in a sense, about your attitude to vaccines and how much you think about this kind of stuff. So go to there, menti.com, and put that code in. We'd appreciate that very much. We'll give you some results in a few moments. But to take us there, Anna, I'm going to come straight to you and jump in with this one, which is that why is it so important that we get this vaccine?
Yeah, so as you know, there's a number of different ways to kind of stop the spread of viruses in society, but one of the most effective ways to do this is with a vaccine. So the reason for this is that when somebody gets a vaccine, it trains your immune system to recognize a foreign pathogen, and then not only does it prevent you from getting the disease, it also then prevents you from spreading it to other people. So this is really the reason we need a vaccine, is so that we can prevent the virus from spreading to more and more people and hopefully squash it within society.
Yeah, and I mean, is this really our only way out of this whole thing? I know we can do test and trace and social distancing and all these things, but really is the vaccine, that's the only way out?
I think it will actually be a combination of a number of different things. So these public safety health measures that we've been putting into place, wearing a mask, social distancing, the test and trace, they're all important. A vaccine is just a component of one of the ways that we're trying to tackle it.
Right. Okay. Now, Paul, my question to you, and treat me like an idiot, and that won't be hard because this is so not my area. I've got an engineering background. So just help me out here. How does a vaccine work? When you nail this, how will a vaccine help to give us immunity against the virus?
So the vaccine, it will produce or be a component of the virus, and it'll have that whenever that enters your body will generate an immune response, a B cell immune response and a T cell immune response against that thing that's injected. And it's that initial generation and the memory of that, which is what makes a vaccine.
Right. So you kind of show the body's immune system something that looks like the virus or is the virus, and the immune system just recognizes it and attacks it when the real virus turns up.
Sure. I mean, traditionally, vaccines have been things like attenuated vaccines, which means like a weakened strain of the vaccine, of the pathogen. For example, things like polio. There are inactivated types where a virus has been killed before being injected. But these are essentially the same as the virus. And though the body is able to see that and react against that and retain the memory of having seen that, and then whenever you actually come infected with the thing in real life, it will mount a robust immune response, a high immune response against that, instead of not having seen it before. If it hadn't seen it before, it has to learn what it is. But if it's already seen it, it doesn't have to learn that, and it can just react immediately.
Yeah, okay. Now, the imperial vaccine, this is really interesting, what you and your colleagues in the Shattock lab are working on, because you mentioned there attenuated viruses. That's not what you're doing in this case. Yours is a more synthetic approach, isn't it? Tell me more.
Ours is completely synthetic. So it's very different from the traditional vaccine modalities, traditional vaccines that people have been given. We use an RNA molecule which enters the cells, and it uses the cells as actually the factory to make protein. This bypasses a lot of complicated manufacturing processes, which usually have to be done outside in large fermenter vats. And this is using your body to make the protein. No virus involved, just the little tiny bit of the virus. No active infection occurs, but because your body makes that, then your body then mounts the immune response and reacts against it, and it creates that memory that you need in order to have a successful vaccine.
So, Anna, nothing like the actual virus goes anywhere near the body. This is the really clever thing, isn't it? So you're kind of, because I've been reading a lot about the spike protein, for instance, on the virus. So that's what binds to the cells in our bodies and causes the illness. And in your case, what you're kind of using the RNA to create these spikes, then to artificially create these spikes, these spike protein-like things, and our immune system says, right, get out of here. Have I summed it up reasonably well? Probably not.
Yeah, exactly. So probably many people have heard of the spike protein now. It's just, if this is the virus, the spike is just a protein on the surface of it. And as you said, that's protein is how it enters cells. So this is an obvious vaccine target for us because that's what we want to prevent, is the virus from entering cells. So as Paul mentioned, we use the cells in your body as our own bioreactor to make the spike protein and train your immune system to recognize that. And then if you are exposed to COVID, then you should be able to neutralize that and prevent you from getting infected.
Right, okay. And we're going to go to the poll results in just a second. But I guess, given that this is a synthetic approach and really getting the body to do a lot of the work for you here, this must make your approach, if you can get this working, really scalable. You can make a lot of this stuff pretty quickly.
Yeah, absolutely. So that's one of the benefits of this, is that it's just really highly scalable. So the dose that we're using in our clinical trial is 1 microgram of RNA, which is just a tiny, tiny amount of RNA. So that means that we can produce about 1 million doses in a liter of our formulation. So it's a very small footprint compared to a normal vaccine production.
Well, that has set us up really well. And you've even made me understand this stuff. So we're off to a brilliant start. And it gets even better because our poll results are in. So thank you very much to all of you out there who have been giving us your views. So let's have a look here. So we basically asked you, how often did you think about vaccines before this whole crisis started? So those who said never thought about vaccines, 1%. Those who said only when I needed to, I think I'd be in that category. So if you're going traveling, you think, oh, I've got to go and get some jabs. Well, 30% said only when they needed to. 45% said sometimes. 20% said often. And 1% said all of the time. So just to sum that up, about just under half of our respondents said they just thought about vaccines sometimes. And I think it's fair to say we're all thinking about them a lot more now. So, and we've had some great questions already coming in on the YouTube chat. So keep them coming. We really appreciate these. And just a quick reminder, of course, this is an event aimed at the general audience, people like me who, you know, we're kind of interested, hopefully vaguely intelligent, but we're not experts. So if you've come here for the really nitty gritty detail about how the pipetting works, we might not satisfy you, but just general questions about what's going on for somebody like me. That would be really, really good. Okay, so let's have a look at this one. This is from Anonymous. We get Anne. Anne is very popular, Anonymous, who asks, do you envisage an issue for elderly or immunocompromised patients getting both the flu vaccine and the COVID-19 vaccine? That's really interesting, isn't it? I've got an 80-year-old mother, and she gets her flu jab every winter. I might be a bit worried about her if she's also taking a COVID-19 vaccine. Who wants to take that?
I can. So yeah, one of the issues. So obviously, you know, people with immunocompromised systems or the elderly are at risk for COVID-19, and so we are actually going to include some elderly people in one of our clinical trials. So typically, as people age, your immune system just gets weaker, and you don't mount such an immune response to vaccines. So this is part of the problem and the reason why more people need to get vaccines, so that you have herd immunity to protect the immunocompromised and elderly. However, because our vaccine, you know, has never been tested in humans before, we don't really know how it will work in immunocompromised or elderly people. So this is something that we plan to test in our clinical trials. So it remains to be seen if we're able to induce an immune response, but that's one of the questions that we're trying to answer.
Right, the whole point of doing the trial. Paul, I'm going to fire this one at you. This is from Alex. Alex says, it's 18 years since SARS. A search for a vaccine has been going on for those 18 years. Why are we any closer to, or probably why are we not any closer to a vaccine for COVID-19 today? So it's a great question, isn't it? You know, SARS was 18 years ago. How come we haven't figured it out yet?
So to put that into context, actually 18 years people have not been researching the SARS vaccine. There was a SARS vaccine for coronavirus 1 that was about to be tested in 2005. And then people stopped becoming infected with SARS coronavirus 1. The interest and, of course, the economics of it meant that then that trial was actually stopped. It wasn't continued. So since 2005, there has been very little active vaccine research in the whole field of coronaviruses. There are a number of coronaviruses which are common cold coronaviruses, but they haven't really been an issue until coronavirus 2 came up. And then whenever coronavirus 2 came up, we used the information from the first bout of research on SARS-1. For about 3 years, there was a lot of research in SARS-1, and we used that, and that really greatly informed our choices and the way that we approached making this vaccine.
Yeah.
So it's not 18 years of continuous research.
Yeah.
It's two short bursts.
Yeah. That's as I remember it. I mean, it's a long time ago now, but I do remember there was the most almighty panic all over the world about SARS. And it kind of, for those of us here outside Southeast Asia, it seemed just to stop. And I guess the vaccine research stopped with it. And I want to put this one to you, Anna. This has been really playing on my mind because so that was SARS-1, which was really contagious. It was a worse disease to have even than COVID-19, but as we heard, it kind of got stopped. So SARS CoV-2, the one we're at now, is really contagious, but thankfully doesn't seem as bad in most cases as the original SARS. Is there a SARS CoV-3 coming down the track that could be both really contagious and also just a really bad illness to have? That's really, and I'm sure it's concerning a lot of our viewers as well. What do you think is the likelihood there, Anna?
Yeah, I think it's actually really hard to say. So obviously, the school of thought at the moment is that these viruses originate in kind of these animal reservoirs, so in bats or other animals, where they continue to evolve and then somehow get passed on to humans. So there's continuing research looking into that and how best to track these viruses as well as prevent them. But it's really hard to say. You know, if we knew what was coming, we would hopefully also be able to prevent it, which we're obviously not able to do yet. So it remains to be seen.
Okay, we'll have to wait and see. Roshan asks, how can vaccines prevent transmission as well as boosting immunity? Such an important question. I guess that's a good one for either of you. Who wants to jump in with that one?
Well, I mean, once you've become vaccinated, we hope that you won't be a vector of disease. You won't be a person that is then able to spread the disease. So you'll become like a roadblock. If someone that's infected comes near you and passes the disease to you, you won't actually become infected and you won't then carry the disease to another person. So that'll affect the chain of transmission. So as well as helping you as an individual, it will help the whole community.
And Anna, I think I was reading with this RNA approach that you're working on that you might end up, is it with more antibodies, but somehow a better immunity compared to if you naturally just got the disease by catching it off someone?
Yeah, so this will be something that we study in the clinical trial. So we're already able to test people in the population that have been infected with COVID-19 and look at their antibody responses. And it's really, there's quite a range to it, but we've already been able to look at that and compare that to how our vaccine is working in animals. But obviously, as our clinical trial goes on, we'll continue to compare how the vaccine response and the antibody response to the vaccine compares to a natural infection. So it remains to be seen how it works in people, but from what we've seen in our preclinical animal studies, it looks like the quantity of antibodies that we induce with the vaccine is greater than the natural infection.
Okay, now a reminder to everybody that we are live on YouTube. Have a look at your clock. If it's 12:47 British Summer Time, you're watching the live stream of this and not the playback later on. So we're inviting your questions, loads of them coming in. I knew we'd get loads on this one. Everyone wants to talk about the vaccine. So we're getting some great questions. So let's go to this one from Louis or Lewis. Will a vaccine eliminate the need for face masks and social distancing?
Will it eliminate the need?
Horrible one to you.
Sure. Again, it's the same idea of transmission. If you are vaccinated and then you don't become infected with the virus, then you won't be able to pass it on to another person. So the idea of face masks is generally that if you're infected and you cough, it'll stop a person near to you becoming infected. So if you don't become infected, then the transmission stops with you.
Right, okay. So when the vaccine comes along, I'm just wondering, will it be this sort of dream, lovely scenario once this gets scaled up and you get your jab and on the way back from the clinic where you've had your jab, Anna, you just say, that's it, I'm throwing away my face mask, I'm going to go to lots of parties, yeah, off we go. Will it be like that?
So there's still a lot of studies that we have to do on this. So part of it will be, does it prevent you from, do you see an antibody response to it? Does it prevent you from getting the disease? And then also, how long does that last for? So we know that it takes a little bit for the vaccine to kick in. You don't immediately have an antibody response. So you can't throw your mask away and go to a party immediately. But I think actually the more important thing is that it will depend on is just in general society, what is the infection rate that we're seeing at the time? I think it's going to be tricky to say, if you get the vaccine, you don't have to wear a mask for this long. I think it will really depend on how much of the virus is still circulating. So hopefully the vaccine is a pathway to there, but I don't think it's a guarantee that you just won't have to wear a mask anymore.
And of course, hopefully testing would have ramped up by then. So we're going to be dealing with better data. And it'll be, I guess, for all, especially you, we're really interested to see what those graphs look like, on the day that the vaccines start getting rolled out, whether just the case numbers just fall off a cliff or if there's a sort of gradual downtail. I guess that's what we're trying to find out.
Yep, absolutely. So yeah, really interested to see what the numbers continue to look like in the future.
Yeah, excellent. Thank you. Now, and just to point out to our audience that Paul and Anna, they're the lab people, so they're not directly hands-on with the clinical trials. You're not seeing the patients involved in the clinical trials. But of course, everyone's interested to hear how the trial is going. I was watching the news in June when your trial launched with its first human patients. So we'll go as far as we can with these questions, but fully understood, you guys, that you're not right there with the people involved with the trials. But Cecile asks, how far are you getting in these clinical trials? When do you think the vaccine could feasibly approved and manufactured? So loads of questions there. I think let's break it down a bit. What are you able to tell us about how far we're into the trial and how it's going?
Yeah, I can start.
Anna, yeah, go for it.
Yeah, so because our vaccine platform, so the self-amplifying RNA, hasn't been tested in humans before, we had to do what's called a dose titration. So the normal dose for the vaccine that we've determined will be 1 microgram, but we can't initially test 1 microgram because the whole goal of the first clinical trial is to look at the safety. So in the first people that got it, we start with 1/10 of the dose and then move up to 1/3 of the dose and then the total dose. So that part of the study, the increasing dose, is complete now. So we've given people the total dose of it, and that seems to be well tolerated. So now we're going on and now injecting or vaccinating everybody with the whole 1 microgram dose. So that's the point we've gotten to so far. So we're just waiting to see how that's going. I think the other 2 parts of the question were when could it be approved and when can it be manufactured? So interestingly, you kind of have to plan for the manufacturing even before you have the approval, just because it does take time and really just more logistical coordination to be able to do that. So we actually have already planned to scale up production in December to make 85 million doses. And the approval will just depend on the clinical trial and how quickly people get enrolled and how fast we get results from that, as well as, the overall results from it. Is it a good vaccine or not? So I think the earliest that we would get an approval or results from the clinical trial would be next year.
Right. Okay. And Paul, let's come back to you to expand that. And that's a great answer to Cecile's 2 questions there, and I really appreciate that. And I guess, Paul, can you put into context just how quick this has been for people thinking, why do we have to wait until January for this? Come on, what do these scientists do all day? Put this into context, just how ridiculously quickly you and many other vaccine teams are working here.
So, I mean, this has been really quick compared to the traditional vaccine discovery, development, and approvals process, which often takes between 5 and 10 years. The reason why it's been so quick is because a number of things, actually. The people were on lockdown, so the only people that were still working on science, we were all working on coronavirus. And there's been a lot of collaboration with a lot of people from different parts of the world. So we've all been working together and working as fast as we can to make this. Also, the platform that we use, the system that we use to make the vaccine is necessarily a very speedy one. It's totally synthetic. So we make the RNA molecule. And we actually made that really, really quickly within a couple of weeks. But it's the whole process of testing it, making sure it's safe, doing the initial assessments before the clinical trial to make sure that it could go into people. And then on top of that, the whole manufacturing and then the rollout. So to get this down into a 6-month to 1-year time period, you can actually see that that's a huge endeavor. And usually people take 5 to 10 years to do this. So this is a complete paradigm shift, complete change in the way people are doing vaccines. And I think it'll become a model for the future.
Yeah, and advances in genetic engineering, of course,
exactly.
that have brought this around. And so if we scoot back then to the beginning, you gave a good account of that process, because Ariana wonders how you even start working on a vaccine. You all turn up to the lab one day saying, well, it's a pandemic, right? We need to crack on with this. What next?
Yeah,
Anna, what happens?
That's a really great question, especially for our type of vaccine. It's kind of interesting. So in January, the sequence of the whole virus was released, and that's really what enabled basically everybody in the world to start making a vaccine for that. So if we didn't have the sequence of it, we wouldn't know what protein or, what the virus even was. So now that we have, really fast sequencing technology, we're able to know the exact genetic code of the virus. And once we had that, we actually got a little bit lucky that this virus is very similar to both SARS-1 and MERS because of this spike protein that we are talking about. So it's actually highly conserved between these different viruses. So we knew the exact protein that was on the surface of the virus. And once we know that target, it's really easy for us we just basically, type out the code of DNA that we need, and then we make RNA from that DNA. So once we have that sequence, that's how we can pivot to making a new vaccine indication.
Right, and that's how you could be so selective then. So you had this genetic blueprint of the virus, you had that very early on in January. And so you were able to go into that and say, right, what's the DNA and RNA within this virus? What's the bit that makes the spikes happen on the protein? And just go for that bit, wasn't it?
Yep, exactly. So we just need the small portion that's on the surface of the virus to make our vaccine. And that's actually what most of the vaccines that are being developed are made to now.
Yeah, okay. I'm just trying to think of some slightly desperate analogy here. And I wonder if it's a bit like, this might be awful, by the way, I haven't rehearsed this. Like you've got the complete works of Shakespeare, a huge amount of information, and you've been asked to give a talk about a particular thing that Shakespeare does, or a particular way that Shakespeare writes. And so you go right into the complete works of Shakespeare and you find one passage from Henry VIII, for instance. You say, right, this really nails what I'm trying to say about Shakespeare. So I'm just going to edit that bit out, and that's the bit I'm going to photocopy and put on the student handout so that they can read it. So we don't need the complete works of Shakespeare every time I wanted to get this particular thing across. How awful. Go and break that metaphor for me. I don't know how it was.
I think that's good. I think in kind of a similar vein of literature, but a slightly different one, I often think of it as like a recipe book. So the whole genome of the virus is like an entire recipe book. And so, there's pages and pages in there, but you don't need all of those other pages if you just want to make one recipe of it. So the spike protein would be like just one recipe. We just need the thing that's on the surface. We don't need the extra, nuclear proteins. We don't need anything else in the virus, just that spike protein. So we take out that page, and that's what we use to make our RNA vaccine.
Oh, I like your recipe metaphor much better. Great stuff. By the way, folks, we still have 15 gorgeous, delicious, wonderful minutes of science to come on this absolutely fascinating topic. So back to the audience questions. And it sounds as if Tanya has had COVID. Tanya says, for those of us with long-term symptoms from COVID-19, could it be dangerous to take a vaccine whilst we still have lingering symptoms? Asks Tanya. So, Paul, let's come back to you because I haven't heard you for a bit.
So, that will be something that will become apparent from the third stage of the trial. But just to apply common sense, you wouldn't really want to take any medical intervention whenever you are currently undergoing some kind of infection. So I would recommend that people in themselves knew whenever they were fully well. And then I think it would be perfectly safe to take the vaccine at that stage. I probably wouldn't recommend, and I probably wouldn't myself, go and have any kind of medical intervention whenever I'm feeling unwell.
Okay, that's really important. So Timothy's got a question about how your approach differs and compares to other approaches, which we kind of did at the top of this video. So probably on that one, I'd say just wind the video back and just watch the first few minutes. I'm not being funny about that at all, but there's a lot to take in, and many of you might want to just go back and watch bits of this again to fully grasp what we're talking about. But as in my layperson's understanding, you're doing this synthetic feat with RNA, where you can make the immune system believe there's lots of this spike protein around and it sends out the immune response. Whereas, for instance, the Oxford group and others are working on attenuated virus, and you show that to the immune system. So that's my very bad way of asking Timothy's question, but I want
the Oxford group are using a different virus to deliver the spike protein. So it's not like it's not an attenuated coronavirus. It's a different virus, which is commonly used to deliver different genes. It's just the adenovirus. It's just another virus that circulates in people.
Yeah, I really appreciate you pointing that out as well. So that gives Timothy a bit of a reply about how the approaches are different. But I want to also expand on Timothy's question to ask you. There's a kind of, and it bothers me a bit, a media portrayal of Imperial and Oxford and the other groups being in this huge competition with each other. It was almost like Man United versus Man City. Anna, it's so not like that, is it? You know, you're all scientists collaborating and working together. It's basically all of you against the virus. Can I typify it like that? I just really want to get that across.
Yeah, absolutely. So I think just, in the news and the media, it's often that all these different scientists and groups are pitted against one another and that it's a race and there's only going to be one winner. But I think our experience has been anything but that, especially in the development of a COVID-19 vaccine. So we actually have an ongoing collaboration with the Oxford group. So looking at, priming with our vaccine and boosting with theirs. And so that's, we've been working quite closely with them and sharing data. So it's actually, we're very friendly with them. But I think that's true, not just with the Oxford group, but really everybody who is developing vaccines and different things right now. So we've gotten a lot of help from other people as far as developing assays, as well as run samples and shared our assay development with other people. So I think actually this has been one of the most open and collaborative times in science that we've seen in recent history, just because at the end of the day, we're all people too. We're all being affected by this virus. And so the ultimate goal isn't to have, our vaccine be the best. It's to have a vaccine that's developed. We just think our platform is a really good way to do that.
And Paul, I guess the overall response will be down to lots of different kinds of vaccines for lots of different kinds of settings.
Possibly. I mean, there's quite a number of vaccines that are being developed, and we would imagine that a number of those will not be successful and will not get to the point where they get licensed for use in people. But if there's 3 or 4 or even 10 that do, I mean, we can use all 10 vaccines and hopefully they will all give equivalent protection.
Yeah, we've got about 10 minutes to go, by the way. Violet asks, will the vaccine need to be repeated over time for protection to be maintained? Are you likely to need booster jabs, I guess, is the other part of that question.
I think that's quite likely.
Yeah, we don't know yet. So that will be part of our clinical trial, is just after people. So the initial schedule is actually that you get a prime and a boost a month later. So people are already getting 2 injections as part of this clinical trial. But we, as part of this study, will follow the volunteers out for a long time and see what their antibody levels are after a certain amount of time. And that will dictate if they need to receive a boost. Is that every year? Is that every 5 years? How long does it last? We're not sure yet, but we are looking into that.
Okay, all part of the trial, all part of what we're trying to find out. And Jan asks, supposing this doesn't work, do you have other ideas for a vaccine? Is there a plan B in your group?
Yeah, there is. We are making a second generation vaccine version, which has got a part in it which will help to enhance the vaccine. We're also going to try and make a vaccine for other parts of the virus. It's likely, though, that the spike protein will be the best target, and so we're going for the best target initially to have the best chance of success. But of course, we have to wait for the trial to finish. We have to wait for how well this performs in people, and then we'll be able to tell whether this works or not. So there is a bit of a plan B, but we've gone for the best thing to start with.
Yeah. So Anna, might that plan B involve going back to what you were doing in January, February? If you had to go back to that initial genetic engineering, that first bit of editing you did, that first bit of the recipe that you snipped out of the recipe book, would you go that far back? Or is this more about all those steps that have had to follow in terms of scaling it and manufacturing it?
Yeah, so actually that's a really great question. So it could go back to, the recipe. Do we need to use a different recipe to have a better vaccine? But there's actually a number of different components to this to this type of vaccine. So I kind of think of it as the plane is off the ground now, right? So it's being tested in humans, and we'll know what sort of immune response we get. But there's different aspects to it. So, does it need to have different wings? So it has a certain formulation to it that we, think we can improve. So that's one thing that we'll be looking at. As Paul said, so changing the RNA slightly, adding another part in it. And there's really many things that we can change, which I think will, come in successive generations of this vaccine. So that's what we'll be going back and looking at, is kind of just tweaking all of the aspects to make sure that we have the most potent vaccine possible.
Yeah. And Amélie asked a really fundamental question here, which is how can you check whether the vaccine's been effective without exposing people to COVID-19? So basically she's asking, is that the only way? You give people the vaccine, send them out into the community, expose them to the virus and hope that it works?
Yeah, so I guess this comes down to, the three different parts and phases of the clinical trial. So the first one is to look at safety, and that's just to make sure that there are, it's well tolerated by people. But then really phase 2 and 3 are characterizing how well the vaccine is working. So phase 2 is looking at immunogenicity. So this is where we measure the amount of antibodies as well as cellular responses in people that have gotten the vaccine. So in the phase 2, we don't expose people to the virus. We're not looking at actually how it prevents the, if it prevents infection. We're just looking at the immune response to the vaccine. So that's more of a way to quantify it. And then in phase 3 is when we look at the efficacy. So that's when we look at, do people that get the vaccine have a lower incidence of getting infected than people who get a placebo? So that is, efficacy and actually looking at prevention of disease is really the ultimate way to qualify whether the vaccine is working. But that's really the third step after we've already confirmed that it's already producing an immune response.
Yeah, and Paul, I had a question for you, and thank goodness Anonymous also asked the same question that I was thinking of, which is, what about if the virus mutates? I wonder how stable COVID-19 is as a virus. So the exact question from Anonymous is, can the vaccine development keep up with the mutation of COVID-19?
So the virus, like all viruses, does mutate. There have been a few noted mutations that have already arisen, but it appears that for the current vaccine candidates under development, that those mutations that are common in different regions in the world haven't affected that these vaccines would work. They haven't affected the potential of the vaccines to neutralize and prevent infection. That's not to say that there won't be a mutation that will do that will prevent the vaccines working, but you can see that because we're able to very quickly change the vaccine backbone, then because we've done all of the clinical trials and the development of the clinical protocol, we should be able to very quickly make a new vaccine that is effective and try and keep up with the virus. Viruses are always mutating. That's how we got here in the first place.
Okay, yeah, so it's all part of, I suppose, understanding and working with viruses. And Jacqueline's wondering, and this is a clinical trial question, so you might not be fully hands-on with this, but let's give it a shot anyway. Jacqueline says, how many people are required for the clinical trials before the vaccine is deemed safe for wider public use?
Yeah, so that's kind of an interesting way to ask the question. So as far as being safe, our initial trial, I guess we are doing a combined phase 1 and 2. So we're looking at both the safety and immunogenicity. And that's in 300 people. Is that right, Paul?
Yes. Yeah.
Okay. So initially, that actually what will qualify as being safe and immunogenic. And then our phase 3 trial will be in a much larger number. So I think it's 6,000 people to look at the efficacy. And so by that point that we are testing it in the general population, we know that it should be safe, although we continue to monitor and look at the volunteers. But then we're really asking the question of, does it prevent disease compared to a placebo vaccine? So it's a little more nuanced than just, safe to distribute, because it also has to be effective. It has to actually have an immune response that it induces.
Yeah, and I'm really getting a sense from this chat about how it's far from straightforward to even be able to say what success would really look like with the vaccine. You know, whether it's number of antibodies, performance versus placebo. You know, I'm really getting a sense. In a way, rather you than me worrying about
Well,
all this. There's so
I
much to
think
think about.
the
Paul.
only criteria essentially will be that phase 3 trial that Anna's talking about. Does it protect people in comparison to a placebo?
Okay.
Is there lower numbers of people that contract the virus in comparison to the control group?
And for trials, do they ideally need to be in places with a high prevalence of cases for you to really know whether
That
it's
would be
working?
ideal.
Yeah. Okay.
I mean, if there's no transmission, there's no way you can tell the difference between the placebo and the infected if there's no infected. So we're not wanting a second wave, but it would be particularly useful at this point.
Right,
but we're also looking at places and partners where there is a higher rate of infection. So that's obviously really necessary to have a phase 3 trial and get results without having to test, thousands and thousands of people. So we're continuing to monitor that situation in the UK as well as partner sites globally.
Yeah.
And I actually saw one of your questioners were asking whether we would deliberately infect people, and the answer is no, we would not. People will become infected naturally, and we're not in the business of putting people at risk.
I'm very glad you said that. It almost didn't need to be said, but I'm still glad that you did. And apologies in advance, by the way, to all of you who've posed questions that we just haven't had time to get to today. It's been a really seriously busy session on here. And as we begin to wrap it up, just from your own point of view, can you tell us what's it like working in this field? Are you sleeping okay? You don't look, you look really well. You're getting on okay. But this is high profile science. Just what's the buzz like in the lab? It sounds like you're thriving on it.
Yeah, I think it's a lot easier for us now. So we work in the lab on kind of the preclinical side, and now that it's moved into the clinic, I think we are having a bit of a break comparatively to how it was in, April, May, doing all of the development and work. It was a lot more hectic then. But I think really for us, it's been a really rewarding experience. And I guess I'll just speak for myself, which is that I think it's actually kind of every scientist's dream to be able to have a clinically translational type of thing like this, right? It's really interesting to make discoveries, but being able to actually impact human health is really the reason for being in this field. And so I just feel extremely lucky and grateful to be able to have worked on this and work in such a good team of people that were able to do it so quickly, but also, potentially have a vaccine that would prevent the spread of this virus.
And Paul, do you think, is this career defining for you? Do you think this is going to be one thing you just look back on and just think, wow,
it's been
what a crazy time that was?
It's been so exciting. I've enjoyed the whole thing. It's what I've been working towards for my entire career, and I'm glad that we are able to do something with the research that we've done and the last 3.5-4 years of actually generating this RNA vaccine platform. This is a great test of that platform, and we're very excited to see the results.
Right, and finally and briefly to you both then, and sticking with you, Paul, what happens next then? Just in a few words, like you have to wait for the trial outcome. Is that next? What's going to happen?
Sure. I mean, the trial outcome is important for this particular vaccine, but we're actually, as I said, more excited about the whole platform, and we will be using this as a template for future vaccines. And we, just before coronavirus, we were developing vaccines against Ebola, Marburg, rabies, flu. You can plug and play with our system. You can put anything into that, and hopefully this is a great test of our system.
Right. So Anna, this is testing the platform, and where next for you then? Maybe trying it with evaluating it with other illnesses, other viruses?
Yep, exactly. So we're, as we talked about a little bit earlier, so we're continuing to look at next generations of the platform. So not necessarily specifically for COVID-19, but just ways in general to make the delivery or the molecular engineering of the RNA better, as well as for other indications. So, we make a number of different vaccines. So for, Ebola, Marburg, rabies viruses. So we're continuing all those projects as well.
All those. The work goes on. Well, Anna Blakeney, thank you so much. And also to you, Paul Mackay. It's been an absolutely fascinating chat. And we'd love to know what you think, dear viewer. We hope you enjoyed it. But you can let us know. There's an evaluation that you can complete. And honestly, it really does help. So please do it. There's a link to that survey in the live chat on the YouTube. You can find out more about these events and other goings on via imperial.ac.uk/what's on, as you might expect. So do join us for future events, and we really appreciate you being here. I'm Gareth Mitchell, signing off and saying goodbye.